Hi everyone, my name is Christina and today I will be discussing the functions of MC1R and more specifically its mutation R67C, which is a specific mutation that causes fur discoloration within mice, like beach mice, but it can also be found within humans and other animals. So MC1R is also known as melanocortin 1 receptor, and it is a G protein coupled receptor found on the surface of melanocytes. Melanocytes is a pigment producing cell found in skin and hair. So when it is activated by a molecule called alpha MSH, it sends a signal back into the cell and it tells it to produce, I believe it's called eumelanin. I hope I'm pronouncing that right, which is a dark pigment that protects the skin and gives it a darker coloration. And that's why MC1R is labeled as a receptor, because its job is to receive signals and then pass them along into the cell. So MC1R is located on the cell membrane of melanocytes. So this is a normal functioning description of my model of MC1R. So like I said, it is found in mice and other organisms like human and other animals. So me creating this model, it has 7 helixes within the membrane. So when the proteins go through, it goes through 7 different phases here, and its mutation is just a slight little change. So we'll talk about that right now. So when you look and zoom in specifically into an MC1R protein, it just means, or specifically the R67C mutation, it just means that the acid arginine in the 67th position gets replaced by cysteine. So arginine is really positively charged, large, and can form strong ionic interactions / bonds, while cysteine on the other hand is neutral, smaller, and it also has a thiol group. So I can show you right now what that would look like. So like I said, this is the normal receptor, what it looks like, the protein, and then when you switch out, this is the placement, the 67th placement, when you switch out the arginine for the cysteine, it's basically like this. So it's basically in that placement where the arginine and the cysteine gets swapped out. So because of that mutation, I can show you one more time. So because of this mutation, MC1R does not work as well because the melanocytes produce less eumelanin and more pheomelanin, which is a lighter pigment. So this leads to lighter colored fur within mice, especially if they're closer to the south and beach areas. So it's more like natural selection to help them survive the wild and survive being placed into natural selection, you know. And so I built my model using Play-Doh, popsicle sticks. So like I said, the Play-Doh is the protein itself within the membrane, and then these popsicle sticks show the membrane as well. So this is like the heads of the phospholipid bilayer. So the head here, and of course inside will be the hydrophobic parts of it. And then, like I say, here is the mutation, and then it just wraps around 7 times, which is usually what a G receptor protein does anyway. So of course This model is definitely simplified and doesn't show the dynamic movement, exact folding, or how complex protein signaling is, but it helps illustrate how one single amino acid change in this protein can affect and create mutations or just change somebody's color. Especially within mice, it changes its fur color, and its fur makes it lighter. So just to summarize this whole protein, so this would be the cysteine that replaces the arginine in the mutation, but it really just makes the protein less active, which produces less eumelanin, and overall just leads to lighter fur, and especially in beach mice, which is what my study was on. It's just reminds us that even a small change at a molecular level creates a big impact on an organism's appearance, livelihood, and environment. So thank you so much. I hope that this video helped understand that MC1R protein works and how mutations like R67C can influence traits like the fur color within beach mice. Thank you so much. Have a good day.